All right, let's dive into one of the biggest questions in all of biology, heredity. For centuries, nobody could figure out how traits were passed down from parents to their kids. It was a total puzzle. And then, a monk in a monastery garden started counting pea plants, and he completely changed science forever.
You've probably thought about it, right? You look in the mirror and you see your mom's eyes looking back at you, but you have your dad's smile. This incredible mix of things you share and things that are different is what makes every single one of us unique. And that's the very puzzle we're going to solve today.
So before we get to the hero of our story, let's just set the scene a little bit. For pretty much all of human history, heredity was a complete black box. Yeah, people knew that kids looked like their parents, but the how and the why, that was all just superstition and a lot of wild guessing.
The field that finally started to answer these questions is called genetics. And it really just boils down to two main ideas. First, there's inheritance. That's the stuff you actually get from your parents. But then there's variation, and that's the reason you're not just a carbon copy. Genetics is all about explaining that perfect balance between the two.
People had all sorts of theories, but that's all they were, just theories. It wasn't until this Augustinian monk, Gregor Mendel, working quietly away in his monastery garden, decided to do something revolutionary. He applied math and super careful observation to the problem. He was the one who finally started to crack the code.
So how in the world did he do it? Well, it all came down to this really brilliant, super methodical, and honestly kind of clever experiment.
Today, we call Mendel the father of genetics, and a huge part of his success was his choice of subject, the humble garden pea. And his reasoning here was just genius. See, pea plants grow fast, they're easy to control, and, this is the most important part, they had these very clear either-or traits. Plus, he didn't just study a few plants, he used massive sample sizes, which meant his results had real statistical power. This wasn't just a lucky guess.
And this just shows you how smart his method was. He zeroed in on 7 specific traits that only had 2 really distinct forms. There was no sort of tall or kind of wrinkled. Nope, plants were either tall or dwarf. Flowers were violet or white. That simplicity was the absolute key to figuring out all the complexity.
And his method was just so meticulous. First, he created what he called pure lines. Basically, he made sure a tall plant would only ever make other tall plants. Then he started cross-pollinating, deliberately mixing a pure tall plant with a pure dwarf plant. And finally, and this was the game changer, he counted everything. The answers, it turned out, were all in the numbers.
And it's in those numbers where the magic really started to happen. Mendel began to see these predictable patterns emerging from what looked like chaos. Patterns that he would eventually turn into the fundamental rules of inheritance.
Okay, so here's the first huge shocker. When he crossed his pure tall plants with his pure dwarf plants, the first generation of offspring, well, they were all tall, every single one. It was like the dwarf trait had just, poof, vanished into thin air. So where did it go?
And this is where it gets really, really cool. Mendel let those all-tall plants from the first generation self-pollinate. And in the next generation, guess what? The dwarf trait popped right back up. And it wasn't random, not at all. He found this incredibly consistent pattern. For every 3 tall plants, there was 1 dwarf plant. A nearly perfect 3-to-1 ratio, time and time again.
So to explain this, Mendel came up with this idea that for every trait, an organism gets 2 factors, 1 from each parent. Today we call these factors alleles. So for the height gene, there's a tall allele and a dwarf allele. The tall one is dominant, which just means if it's there, the plant's going to be tall. The dwarf one is recessive, which means it can be hidden by the dominant one.
And that simple idea led to his first 2 groundbreaking laws. The law of dominance, that's what explains why all those first-generation plants were tall. The dominant tall allele was just hiding the recessive dwarf one. Then you have the law of segregation, which explains that 3-to-1 ratio. It basically says that when a plant makes its reproductive cells, its pollen or eggs, it's 2 alleles for a trait split up. So each egg or sperm gets only 1, totally at random.
But you know how it is, right? Biology's never that simple. Mendel's rules were the foundation, for sure, but scientists who came after him found out that sometimes the rules get a little complicated.
So Mendel's peas showed what we now call complete dominance. One allele completely masks the other. But in other plants, like snapdragons, you find what's called incomplete dominance. In this case, neither allele is totally dominant. So if you cross a red flower with a white one, you don't get red. You get a beautiful blend of pink flower.
And then there's another twist, codominance. This is where both alleles show up, fully and equally, at the same time. The perfect example is right inside of us, our ABO blood type. The alleles for type A blood and type B blood are codominant. So if you get one of each from your parents, you don't get a blend. You get type AB blood, where your cells are expressing both A and B traits. Now, what's so amazing is that Mendel figured all this out just with abstract rules. He had no clue what the physical thing was inside the plant that was doing all this. His factors were just an idea. It would take decades for technology to catch up and show us the physical basis for his laws.
And the timeline here is just wild. Mendel publishes his incredible work way back in 1866, and nothing. Crickets. It was just too far ahead of its time for anyone to understand. It wasn't until 1900, long after scientists could actually see these things called chromosomes under a microscope, that his work was rediscovered and everyone had this collective aha moment.
And here it is, the big connection. Scientists realized that Mendel's abstract factors were real physical things. They were genes. And those genes were located at specific spots on those chromosomes that they were seeing inside of cells.
The breakthrough insight from two scientists named Sutton and Bovary was that the way chromosomes moved around during cell division perfectly matched what Mendel's factors were doing. Chromosomes came in pairs, they split up into reproductive cells. It was a perfect match. Mendel's math finally had a physical home.
So, okay, why does a monk from the 19th century and his pea plants still matter to us right now? Well, because these basic rules he figured out are the absolute bedrock of all modern genetics and medicine.
Mendel's laws are what allow us to understand and even predict how certain genetic disorders are passed down through families. Conditions like sickle cell anemia or hemophilia or muscular dystrophy, these are called Mendelian disorders because they're caused by a mutation in a single gene, and they follow his rules of dominance and segregation.
And that means these conditions, which are caused by these tiny changes in our genetic code, aren't just random bolts from the blue. Thanks to Mendel, we can trace how they're inherited through a family tree, we can understand the odds of them being passed on, and that knowledge is just incredibly powerful for doctors, for researchers, and for families all over the world.
So, from a simple garden experiment, we got the key to understanding our own biological blueprint. It's just an amazing reminder that sometimes the biggest discoveries come from the most humble places. And it leaves us with this really exciting question. With all the tools we have today, what secrets are we going to unlock next?